So the reproductive system, right? Let's go ahead and take a look at the reproductive system. We're going to go ahead and get started with two things. So the lecture today will focus on meiosis, the role of meiosis in the production of gametes. Recall that gametes are sex cells, okay, sperm and egg. That's what gametes are. So meiosis has a role in gamete production. And then from there, we'll move on to the male reproductive anatomy. And then we'll go ahead and end with the female reproductive anatomy. No physiology today, just anatomy in this lecture. All right, so starting off, meiosis. This is something you saw back in biology, right? Back in regular biology. It's been a long time, I know. So let's recap real quick. Recall that our cells are eukaryotic cells. Okay, so human cells have 46 chromosomes each. They are diploid cells, meaning that they have two sets of chromosomes rather. Okay, the half of them come from dad, so paternal chromosomes, and the other half come from mom, maternal. All right, so that makes it 23 and 23 pairs from each one. Alrighty, so again, just wanted to highlight those, the fact that, again, half chromosomes come from mom, half of them come from dad. These are the parts of chromosomes real quick. For example, the centromere is the middle of each chromosome. Each side of the chromosome is called a sister chromatid, if you recall. And then there's paired homologous chromosomes here, which are very similar chromosomes, one from mom, one from dad, and meaning they have the same genes, right? Or similar genes. And sure enough, right, there it is. Some terms from that you learned back in biology to highlight some of the important things from chromosomes. Now, during meiosis, again, recall meiosis is in essence the formation of gametes, which are sex cells. Meiosis takes place in 2 rounds of division. So 2 rounds of division, not to be confused with mitosis. That's different. All right, meiosis, even though it has the same phases, if you recall, prophase, metaphase, anaphase, telophase, there are 2 rounds of division here. All right, therefore, the end result is going to be, obviously, it's going to be different than mitosis. All right, so let's forget about mitosis right now and let's just focus on meiosis. So here we have meiosis 1. Okay, the first stage. We have a cell that just ended the cell cycle, right? The cell just ended the cell cycle. It entered the division stage of meiosis, right? So the first division is called meiosis 1. The reason why this is important is because during prophase of meiosis 1, there is a combination, there's a mixing of genes within chromosomes. So the different chromosomes, they not only pair up together, but genes from one chromosome cross over to the other chromosome, right? So they mix up right there. We know this as genetic variation. Right, this is where we achieve genetic variation, and that's the reason why we all look different. Okay, that's the reason why we all look different. We behave differently, we interact differently with the environment. Even if you have an identical twin, right? You're not exactly the same as your identical twin. Your genes are exactly the same, but nevertheless, the environment has a different effect on your phenotype, on your characteristics, if you will. All right, so this is the reason why. This is one of the main reasons why that's the case, crossing over. Okay, so this is arguably one of the most important phases of meiosis within all of the phases that there are in mitosis, out of the 8 to 10 phases. Next is metaphase, chromosomes align in the middle of the cell. Next is anaphase, chromosomes are separated. Half go into a pole of the cell, the other half go to the other pole. And then telophase, makes 2 new cells. So at the end of meiosis 1, we have 2 haploid cells, meaning that these cells are going to have 23 chromosomes, 23 pairs of chromosomes each. All right, but they're still considered to be haploid. So that's meiosis 1 in a nutshell. Meiosis 2. Meiosis 2 is another round of the same phases, prophase, metaphase, anaphase, telophase. It's essentially the same, with the exception of crossing over. In meiosis 2, you don't have any more crossing over. It's just pairing of the chromosomes in preparation for metaphase, which prepares the chromosomes for anaphase, which prepares them for telophase, which makes another 2 new cells from each parent cell. So the result of meiosis 2 is 4 haploid cells with half the amount of chromosomes as the original parent cell. Therefore, these cells right here are going to have 23 chromosomes each. 23 chromosomes each. Okay, and here's why that's important, because egg cells and sperm cells contain 23 chromosomes. They cannot have more than that. Why is that the case? Well, because when they come together in fertilization, 23 plus 23 makes a 46 chromosome zygote, which is in essence what a human is, right? So if sperm or egg have more chromosomes or less, then obviously that's going to have an effect on the viability of the baby.
Gametogenesis. Gametogenesis is the process of formation of gametes. The primary sex organs, the testes in the males, produce sperms, and ovaries in the females produce ovum. Gametogenesis is further classified into 1. Spermatogenesis. 2. Oogenesis. Spermatogenesis begins at puberty. Significant increase in the secretion of gonadotropin releasing hormone, which stimulates the pituitary glands and stimulates the secretion of 2 gonadotropins, luteinizing hormone LH and follicle stimulating hormone FSH. LH acts on the Leydig cells and stimulates synthesis and secretion of androgen. Androgen in turn stimulates process of spermatogenesis. FSH acts on the Sertoli cells and stimulates process of spermatogenesis. Immature male germ cells, spermatogonia, produce sperms by spermatogenesis. Each spermatogonium is diploid, which divide by repeated division containing 46 chromosomes. Spermatogoniums present on the inner side of seminiferous tubule multiply by mite division, forming primary spermatocyte. Primary spermatocyte undergo meiotic or reduction division, forming secondary spermatocyte. Secondary spermatocyte undergo second meiotic division to produce 4 equal haploid spermatids. 4 equal haploid spermatids transform into spermatozoa, sperms, by a process called spermiogenesis. Sperms moved from seminiferous tubules by process of spermiation. The seminal plasma, along with the sperms, constitute the semen. The functional sex accessory ducts and glands are by testicular hormones, androgens. Structure of a sperm. It is a microscopic structure composed of a head, sperm head contains an elongated haploid nucleus, the anterior portion of which is covered by a cap-like structure called acrosome. The acrosome is filled with enzymes that help fertilization of ovum. The middle piece possesses numerous mitochondria which produce energy for the movement of tail that facilitates sperm motility essential for fertilization. The human male ejaculates 200 to 300 million sperms during coitus. 60% must have normal shape and 40% show vigorous motility. Oogenesis. The process of oogenesis involves development of an ovum which starts from a primordial oocyte around which 2 to 3 layers of cells from the ovarian stroma. The outer layer is fibrous and known as theca externa. An inner layer, which is somewhat vascular and cellular, is theca interna. The cellular mass is soon separated into two parts by the collection of fluid, the liquor folliculi near its center. A homologous membrane, the zona pellucida, appears between the primary oocyte and the follicular cells. These cells increase rapidly, forming many layers. The theca layer are differentiated and the follicle assumes a deeper position in substance of the ovary. The primary oocyte divides meiotically, giving off the first polar body and becomes a secondary oocyte. The germ cells become mature ovum after the second polar body is released. During later stages, the fluid-filled spaces, the atrium appears in the membrane granulosa layer formed from the cells separated off by the liquor folliculi. The resulting structure becomes a vascular or graphene follicle. As the follicle matures or ripens, it projects out from the ovarian surface as a small cyst-like oval swelling which bursts to liberate the ovum, the process being called ovulation.
Yeah. So again, gametogenesis, gameto means gametes, which is in essence sex cells. Now, during the physiology day, which is going to be next time we meet, we'll go over all those processes, all those details. For now, I want to focus on the anatomy. So starting off with the internal organs of the male. Keep in mind that this is divided up into internal and external anatomy. So looking at starting anteriorly and going posteriorly here, starting off with the penis evidently. Okay, so penis contains a duct inside of it, right, called the urethra, as you probably know. The urethra extends all the way in, right, so it extends deep up into the bladder, essentially, but there is a connection, there's a bifurcation right by the location of the prostate here, which again connects to the ejaculatory duct. Okay, this ejaculatory duct is going to go and emerge from all of the duct system that comes out of the testes. Okay, so this duct system right here originates in the testes and in this region called the epididymis, which is connected to the actual testes themselves. And inside of the testes, evidently, is where sperm is generated. Okay, the testes contain many different types of cells, one of which creates sperm cells. So we'll go over that in a minute. So the penis itself is the copulatory organ for males. So again, copulatory meaning mating, sex, if you will. So the composition of it is spongy connective tissue, which can be filled with blood. Okay, so not only blood, but we actually release a chemical, nitric oxide, which helps in dilating the arteries, not only of the penis, but the arteries everywhere throughout the body. it dilates the arteries and it allows for, for the free flow, a better flow of blood, right? So during sexual stimulation, there is a higher release of nitric oxide, which in turn increases the flow of blood into the penis, which is in turn going to go ahead and fill the penis with blood, causing an erection, evidently. All right, but that's again part of the sexual stimulation, which is more of the physiology part. So I'll get back to that. The urinary bladder is part of the urinary system, so we're not going to talk about that right now, even though it's indirectly connected to the urethra. So therefore, in males, the urethra has two jobs, right? In males, the urethra has two jobs. One is urination, but the other one is ejaculation, or ejaculation of semen. Now, sperm is made, as you know, in the testes, and it's accumulated in the epididymis, which is an extensive set of tubes, essentially. It's a duct system that connects to the ductus deferens, so this long tube right here. It connects over to the accessory glands to include the seminal vesicle here in blue. It's not blue, evidently. It's just so that we can differentiate it. The prostate gland right here. And those two connect with the ductus deferens to form the ejaculatory duct, which is right there. The ejaculatory duct goes ahead and connects to the urethra right at the level, as mentioned before, of the prostate. Here's the prostate right there. What the prostate is going to do is it's going to go ahead and release seminal fluids. It's going to release fluids that mix with the sperm that came from the epididymis. And that mix, right, that blend becomes what is known as semen. Okay, so we'll talk more details about it next time in physiology. So if there is sexual stimulation, then semen can be made to go ahead and be forced out of the penis, evidently in a process known as ejaculation. But nevertheless, the formation of it occurs right here, right, with the seminal vesicle contributing to it, the prostate contributing to it. And their contributions are not only water, fluid contributions, but also enzymes, proteins, as well as other molecules that are important for the viability of sperm once the sperm is ejaculated into the vagina, and those molecules are going to help sperm swim up into the cervix and then later into the uterus. And then some of those are going to hopefully make it into the fallopian tubes to allow for fertilization to take place. Okay, so let's go ahead and zoom in to the testes. So the testes are found evidently outside of the body. All right, so they would be not the same thing, obviously, but the equivalent to the ovaries. Okay, the equivalent to the ovaries in females. Now, the ovaries, as you know, they're found in the body, right? They're found inside of the pelvic cavity, not the testes. The testes need to be outside. The reason why is that they require a colder temperature. They require a colder temperature to allow for the formation of sperm, okay? To allow for meiosis to go ahead and form sperm through the respective process known as spermatogenesis. Okay, I'll come back to spermatogenesis next time. Now, if we zoom in to the testes, we're going to find that the testes are divided up into lobules. Okay, so these oval-shaped structures right there, they're called lobules. Lobules contain seminiferous tubules inside of them. This is the spelling, right? Seminiferous tubules inside. There's millions of them inside of each testis. And millions of seminiferous tubules, all of which are interconnected, and they lead over to the rete testis, which is this complex network of tubules. Right, so the retate testes are extensions of the seminiferous tubules. Now, this network right here is going to go ahead and extend into the ductus epididymis, and there's three places to the ductus epididymis. There's a head, a body, and a tail. Starting with the head. The head is essentially the start of the epididymis, which, don't forget, it's part of the duct system, right, of sperm production. Within the seminiferous tubules, if you are able to take a cross section of a seminiferous tubules, you are going to find certain types of cells, to include sustentacular cells and interstitial cells, both of which are involved in not only supporting the seminiferous tubules, but in generating the sperm cells, right? That the males are essentially in charge of, and that's their contribution to reproduction, right? So interstitial cells have other functions, for example, releasing testosterone, for example, to go ahead and nourish these structures here. Because without the presence of testosterone, sperm production cannot take place. Testosterone is a very important molecule, as you probably know, in the production of sperm. All right, so these, again, sustentacular cells, they become sperm cells eventually with the presence of testosterone, and they go ahead and develop into sperm cells. Now, once the sperm cells, which you can see right there in the middle, once they're formed, they go ahead and swim outwards into the retate testes, into this network right here, and then they continue on their journey into the head of the epididymis. They move on to the body of the epididymis, continue to the tail of the epididymis, and then up into the ductus deferens. Right? And this structure right here is going to go ahead and hold on to these sperm cells. It's going to store them for a period of up to 6 months. Okay, so in case there's no ejaculation, the sperm cells can be stored in the epididymis for a few months. After which time, sperm cells can, simply be recycled, right? So at the end of a few months, those cells that are not ejaculated, they become inviable, and then they're simply just recycled. Right? They die, and they're essentially made into their constituents, and they're made again into other sperm cells. Okay, so if there is if there's sexual stimulation, right, then sperm cells are going to go ahead and travel through the ductus deferens up into the spermatic cord, which contains not only the ductus deferens, but the testicular arteries and veins necessary to nourish and drain the testes, right, of blood. Now, with that said, in cases where males don't want to procreate, right, they can have their ductus deferens severed, right? So they can simply go to the doctor. The doctor can go ahead and right there in the office, without making the man go to sleep, if you will, right? Just by applying local anesthesia, they can go ahead and sever the ductus deferens and prevent sperm from flowing from the epididymis into the prostate, right? So that's a form of preventing pregnancy, right? So now, can that be reversed? Yes, it can. Okay, and we'll go over details of that next time. All right, so Looking at the same structures, but in another view. Again, starting with the external, which is obviously the penis, and the testes are also externally, right? And they're found within a scrotum, which is a sac-like structure, evidently that has skin on the outside, but most importantly, there's muscle that surrounds the scrotum, which in essence carries the testes. So I'll talk more about that in a bit. The penis right here is divided up into several sections: the glans penis, the body of the penis, and then the root of the penis. The rest of it is divided up into corpora cavernosa, which again is in corpus or corpus spongiosum, which is spongy tissue that can be filled with blood. Okay, so now running through the middle of it, but more inferior, is the actual urethra itself. There's also these bulbourethral glands right here that also contribute to secretions, right, in the production of semen. Moving up, here's the prostate. Here's the ejaculatory duct with the seminal vesicles on both sides. Ductus deferens connecting over to the epididymis, which connects to the testes. Here's a cross section of the penis. So corpora cavernosa, one on each side. On top, there's the arteries that are necessary to, again, nourish the tissue, but also drain deoxygenated blood from the tissue. Right underneath, located inferiorly, is the spongy urethra. Right? So there's the urethra right in the middle. And that is surrounded by corpus spongiosum, which is the same tissue that the corpora cavernosum is made of. Again, spongy tissue. So here's yet another view. This shows the inguinal region here. So the inguinal region is the transition between the abdominal cavity and the pelvic cavity. That's what the inguinal region is going to go ahead and do. It's filled with muscles and ligaments and tendons that are necessary to go ahead and keep everything in place, essentially, right? So keep everything from moving, attaching, anchoring. But then most importantly, what I wanted to mention is these muscles right here, the cremaster muscle and the dartos muscle. Cremaster muscle is deeper in relation to the dartos muscle. They have two jobs. One is contraction of the cremaster muscle is actually going to go ahead and shorten the extension of the testes downwards. Okay, so if this muscle contracts, then the testes are going to tend to move inwards into the body, closer to the body. The relaxation of the cremaster muscle is actually going to allow extension of the scrotum, therefore allowing the testes to extend outward and away from the body. All right, so therefore, if it's hot, what's going to happen is that there's going to be relaxation, therefore allowing for testes to move downwards. If the body's hot. If it's cold, then the opposite is going to take place. The cremaster muscle is going to go ahead and contract on both sides, therefore allowing the testes to move inwards, very close to the body. All right, now the other muscle right here is going to go ahead and allow for the surface area of the skin surrounding the scrotum to either increase or decrease. Right, so if it's too hot in the environment, right? If it's way too hot, then what you want to do is you want to increase the surface area of the skin. Therefore, the muscle relaxes. That increases the surface area and allows for sweat to diffuse, right? For that heat to diffuse out in the form of sweat, evidently. If it's too cold, then what you want to do is you want to reduce the surface area. Therefore, the dartos contracts, and that reduces, wrinkles up the skin around the scrotum, if you will, reducing therefore the surface area so that not a lot of heat is lost to the environment. All right, and you can read this on your own later. That's a summary of the parts. We'll talk about physiology next time. Let's go into anatomy of the female. So again, external, internal anatomy. Externally, we call it vulvar region. Right, so the vulvar region, I'll get to it in a bit. Going in to the vaginal canal, ending at the cervix, and then entering the uterus, following in the fallopian tubes, and ending with the ovaries, evidently. All right. So now notice everything else, right? So these structures have to be held in place as well. So there's a need for ligaments, tendons, right, to go ahead and anchor and attach everything to the pelvic cavity. To the pelvic cavity. Notice the peritoneum. We talked somewhat about the peritoneum, actually extensively in the beginning of last semester. This would be one of the membranes, one of the cavities of the body, right? One of the major cavities of the body. The peritoneum touches the superior aspect of both the male and the female reproductive system, but these are not found within the peritoneum. It just touches them. Okay, so urinary bladder is, notice the difference between male and female bladder. The female bladder is located anteriorly to the vagina, anteriorly. Therefore, the urethra does not merge with anything else within the vagina. They're two separate openings. Right? Here's the vaginal opening, here's the urethral opening in females. Okay, in males it's different, right? Males, the urethra merges with the ejaculatory duct. Therefore, they're ending up in just one urethra, right? That has two functions, urinary and ejaculatory. Not in females. In females, it's different. Okay, so here's a version with the pelvic bones, right? So here's the pelvic bone, the coxal bone on the left, the coxal bone on the right, sacral bone right here towards the back, the posterior aspect. Pubis symphysis right here, which is made up of cartilage. And then the bladder, which is right below the uterus. The uterus sits right above the bladder, right? And then you can see the fallopian tubes or uterine tubes right here. Same thing. Ovaries, again, all held in place with the respective ovarian ligaments, as well as the round ligament, which is a broader ligament holding on to the entire structure right there. Okay, so if we zoom in here to the uterus, starting at the bottom of the uterus. So here's the cervix. This is the opening into the uterus. That was one of the questions in the opening. The cervix is the ending of the vaginal canal. Okay, this is the end of the vaginal canal. Notice how the vaginal canal is actually lined by these folds. These are called rugae, right? So that's how you spell that. These folds have a stimulatory effect during copulation, during sex. So they have a role, essentially. Again, the cervix is located right at the end of the vagina, so superiorly on the vagina and inferiorly on the uterus. It's the opening to the uterus, which is this entire region right here. The uterus is lined by endometrium, right? So this is the endometrium of the uterus, followed by the myometrium and the perimetrium. The endometrium is the one that is shed at the end of the menstrual cycle, which differs. On average, it's a 28-day, but it differs in females. So this is the cavity of the uterus right here. This is where the baby forms. The sperm fertilizes the egg inside of the fallopian tubes. The zygote, if fertilized, evidently, travels over to the uterus, implants on the endometrium, and forms into a baby, and that we call that pregnancy, right? And that lasts anywhere from 36 to 40 weeks. So fallopian tubes, ovaries, right here, with the medulla of the ovaries, which is the deep side, and the cortex, which is the superficial side. This is where the egg is formed, the ova, right? This is where the egg is formed throughout the first part of the menstrual cycle. Okay, so the first 14 days essentially form the egg. The 14th day releases the egg. That's called ovulation, and then the last 2 weeks is basically just waiting for sperm to fertilize that egg. The egg is released in the fallopian tube, and this is mostly where fertilization takes place, either on the left side or on the right side. Okay, so moving into the external genitalia. Again, this part is known as the vulvar region. I don't think I have it here. It's spelled V-A-L or V-U-L, I'm sorry, V-A-R, V-U-L-V-A-R. Vulvar region. Okay, externally. So starting superiorly here, there is the clitoris, which mostly has stimulatory effects. Right under it is the urethral orifice. So this is for urination, evidently. And then there's the vaginal orifice right here. So all of it is covered and protected by labia, 2 sets of labia minora, labia majora. Now, the region that includes the anus, all of this entire region is known as the perineum, and it's the same in males. Obviously, males, you'll see the testes here, the scrotum, and then a region of tissue, and then a region of tissue right about here, and then the anus. So this entire region again, perineum. Now, the perineum includes the anus, right? But the vulvar region does not. It's just the external female genitalia. Mammary glands, these evidently include the breasts. Notice how the breasts are mostly made of adipose tissue. Recall adipose is fat. Okay, this is fatty tissue right here, which is made of adipocytes. Adipocytes are fat cells. They're cells filled with fat. Okay, so that's what they're made of essentially. Now, the fatty tissue, the adipose tissue, forms the actual network, right? The actual framework of the breast. But then it contains all of these tubules, all of these ducts right here. These are the lactiferous ducts. Excuse me. Which are also divided up into lobules, and these lobules have a role, and their role is to produce milk. Okay, so they're stimulated by hormones, but again, that's physiology. So we'll come back to physiology and milk production. All right, so that's a summary of the female genitalia. And